Gonna start with. I just want to say thanks for joining us today. Today we probably won't be speaking specifically about PDE, but more so about your experience working through the gene editing clinical trials, I believe, right, for AADC. Did I get that right?
Right.
Yeah. And I think the goal is really for us, the collective us, and anyone who watches this to understand how some of these things look in more or less practice or more or less reality and take it out of the hypothetical or the scientific, which is where I know I live a lot in the scientific and the sort of, research oriented mindset, and I haven't had experience in the so much translational world of it, and I know that you have, and it's very kind of you to share that experience with us. So uh, kick it off, take it away, and hopefully um, everything will work. Let me know if all of the sharing and all that stuff works out.
Okay. Well, thanks, Anna.
Okay.
And Curtis and everyone on the screen. We decided I was going to talk about our experience with gene therapy for AADC deficiency. So, like you said, it's quite different than PDE. Um, but there should be parallels, right? And I was kind of wondering if Curtis had an update about our grant submission. Last I heard is the study section meeting was postponed until further notice.
Yeah, still true as of this morning, and I did have a conversation with the program officer.
Well, so this will address, and it won't, I don't remember exactly how long you want to allocate to the talk, but it doesn't have to be very long.
That's
It
fine.
won't be it won't be a whole hour, but
Mhm.
this is about our experience using gene therapy for aromatic amino acid decarboxylase deficiency. And I have Skellig Stone on the slide. He's our neurosurgeon who became involved with this several years ago when I kind of referred him to go on a trip to Taiwan to see what they were doing there, since there's a gene founder effect, so there are more patients there. And he actually contributed some important ideas about using a clear point catheter system, which he thought technically would be the ideal system. And that led to a study that we got involved with gene therapy that ultimately led to FDA approval for the gene therapy and the catheter system. So the biochemistry of this deficiency is shown on this slide. Back, at least when I was in med school, I'm showing my age, I learned this enzyme here is dopa decarboxylase. And by the way, B6 is a cofactor as it is for all decarboxylases. It turns out this enzyme's name was changed from dopa decarboxylase to aromatic amino acid decarboxylase because it not only converts L-dopa to dopamine, but it converts 5-hydroxytryptophan to 5-hydroxytryptamine, which is serotonin. And of course, these are major neurotransmitters, and dopamine gets converted into the catecholamines norepinephrine and epinephrine. And it really all starts from phenylalanine to tyrosine to L-dopa and tryptophan to 5-hydroxytryptophan, and it involves the conversion of tetrahydrobiopterin to dihydrobiopterin. And that begins from guanosine triphosphate. The first enzyme that breaks that down is GTP cyclohydrolase 1, which is deficient in Sagawa disease, dopa-responsive dystonia. So we're really in the realm of movement disorders, although these patients have cognitive impairment, some have epilepsy. But the predominant issue, as you might expect, is these patients basically have congenital Parkinson disease, where instead of the substantia nigra in the midbrain degenerating, the enzyme's not there to produce dopamine. And dopamine cannot therefore go to all the various structures it's supposed to go to, especially the basal ganglia. This disease was first described by Keith Hyland and Peter Clayton, Of course, Keith now runs the Medical Neurogenetics Laboratory in Atlanta, and Peter Clayton is of tremendous fame at the Great Ormond Street Hospital, University College London. They described this in 1990, when I first started my career, as an autosomal recessive deficiency of the enzyme, which causes combined serotonin and dopamine, epinephrine, norepinephrine, or catecholamine deficiencies. And the CSF was really the mainstay of diagnosis, where there's nothing really peripheral that you could use to make the diagnosis, kind of like B6 dependency. I mean, there's no blood test. You'd have to really do a CSF for a P5P level. Same with this. We relied on CSF neurotransmitters, looking for a low homovanillic acid and low 5-hydroxyindoleacetic acid, which are the major metabolites of dopamine and serotonin, respectively. These patients also had high L-DOPA levels, 5-hydroxytryptophan levels, and 3-O-methyl-DOPA levels, and normal pterins to make the diagnosis. And the clinical features are predominantly hypotonia in infancy with a pretty significant paroxysmal movement disorder. Lots of oculogyric crises where the eyes get stuck, usually up into the side for minutes to hours. It seems very uncomfortable. But the other movement disorders, including torticollis or twisted neck, a lot of dystonia of the limbs, sometimes flexor spasms, which would be confused with infantile spasms, which is a form of epilepsy, myoclonus, which is subcortical, tremor, etc. Also, they get autonomic symptoms, again, with the catecholamine deficiency, like temperature instability, sleep disturbance, irritability, developmental delay. A lot of them get really bad nasal congestion, stuffy nose. Is actually, the funny thing is the parents relate to that more than anything. And hypoglycemia, they can really have a deficient gluconeogenic response to stress and get very seriously hypoglycemic, which can lead to crises. Now, it tends to present in the neonatal period, or I really find kind of early infantile period in the first, say, 4 to 5 months of life. But it can present as a neonate with hypothermia, lethargy, poor sucking, ptosis, and hypotension. Almost all of these patients are misdiagnosed with cerebral palsy or epilepsy or mito, sometimes myasthenia, because there is a fluctuation to it, or hyperexplexia, the startle syndrome, where you do a nose tap test and a baby, flips off the bed. They get dysautonomic problems. Imaging may show some progressive atrophy, but otherwise is pretty nonspecific or unremarkable. The mainstay of treatment for years has been dopaminergic agents. Like I use a lot of pramipexole or ropinirole in these patients. Anti-epileptic drugs or anti-seizure medicines. MAO inhibitors I find very useful, monoamine oxidase inhibitors, to increase the levels of the catecholamines. Anticholinergics sometimes and serotoninergics aren't really that helpful, but frankly, the serotonin line is much less affected than the dopamine line of synthesis. And the HVA levels are way lower than the 5-HIAA levels. Outcome's pretty poor. We used to say there was a variant in girls. They did, I can't even remember. I'm forgetting now. I think they did worse, but that's kind of gone away. Now, things changed dramatically, of course, with the mapping of the human genome, and all of a sudden gene sequencing became available, and some companies got very interested in this disease, and they started adding this gene to panels of children who are being studied for cerebral palsy or movement disorders. And cases, and back in the day in the 90s when I was starting in the early 2000s, we were diagnosing these patients with CSF neurotransmitters, which is a difficult test to do. It has to be a perfect tap, like a champagne tap, with really no blood. It had to go on special tubes in dry ice immediately and get sent to a very specialized lab. There are only 2 laboratories in the United States I'm aware of that measures CSF neurotransmitters. That's Medical Neurogenetics Lab and the Institute for Metabolic Disease Lab in Dallas, which is part of Baylor University. It's not the same as Baylor College of Medicine in Houston. But that changed with the genetic sequencing of the DDC gene, dopa decarboxylase gene. And then a company got very involved. It's currently PTC. PTC actually bought out the first company I'll be honest with you, I can't remember the name of the first company that got involved, but because I was involved in neurotransmitter disease research, really because of my work in SSADH deficiency in the GABA line, but because of that, I was going to meetings of the PNDA, the Pediatric Neurotransmitter Disease Association. I got to know a lot of the investigators and became friendly with them, like Keith Hyland and Nenad Blau. And I ended up seeing a fair amount of kids with this disorder as well as related disorders like tyrosine hydroxylase deficiency, or people with Sagawa disease, many of them being adults actually, or autosomal recessive GTP cyclohydrolase 1 deficiency, which is a really severe disease, or sepiapterin reductase deficiency. But anyway, so I ended up getting involved in this disease as well as SSADHD. And for example, we had a meeting in San Servolo, which is this beautiful island right off the coast of Venice, Italy. And this consensus guideline came out in like 2017. The meeting was in 15, I think, or 16, led by Tessa Wassenberg and Thomas Opladen in Heidelberg. And we had consensus guidelines just like Curtis did for PDE. This was done for AADC, very similar process. And then gene therapy started to emerge. And it really started with the Taiwanese because they had the most cases. And there were also a lot of cases in Japan. And this important paper got published in Brain. Gene therapy improves motor and mental function of AADC deficiency, 2019. So this is pretty recent stuff. This was maybe, about 5 years ago. And then, as I said, Skellig Stone, he's really our epilepsy surgeon. He's a fantastic neurosurgeon who has a lot of experience with the DBS, deep brain stimulator, and that kind of intervention of devices for, say, movement disorders. So he went to Taiwan and observed what they were doing there, and he recommended this SmartFlow MR compatible cannula, which was studied in the US, basically at 3 children's hospitals that I know of: Texas Children's, which did the most cases, Cincinnati Children's, and Boston Children's. There may have been cases of Colorado Children's, but I actually am not certain about that. Anyway, the gene product is called Eladocagene exuparvovec. I don't know where they came up with these names. And so this was the study that we got involved with, and that led to FDA approval of gene therapy this past November of 2024. So this was an open label study of the gene therapy, which was given in a AAV virus, so it like an attenuated adenoviral. As a lot of you will remember, back in the early 90s, the University of Pennsylvania did gene therapy in a young man named Jesse Gelsinger with pretty mild ornithine transcarbamylase deficiency, but he was old enough that he could give consent, which the ethicist thought was important. I think it was a really bad idea, as to a lot of people, because it's not like he needed it to survive. But anyway, he got the adenoviral mediated gene therapy and died of viral hepatitis. That put gene therapy behind by 20 years, people have said. But fast forward 20 years, and there are alternative ways of doing it. I have a whole separate talk on that. But the most common is to use an AAV, an attenuated adenoviral vector, to carry the gene therapy. And that's what we did in this study. But interestingly, this gene therapy is not given intravenously or even intracisternally or intrathecally in the spinal fluid space or intraventricularly. It's given directly into the brain. It's given into the main target organ for dopamine, which is the putamina. So in this coronal view, in this sagittal view, and this axial view, you can see the patient's MRI, and you can see that the catheter is used to inject the gene therapy in both the anterior and the posterior aspects of the putamen, really the largest of the basal ganglia. Here you've got the head of the caudate, you got the globus pallidus, and the putamen. There's also subthalamic nucleus, which is kind of hard to see, and that's very small. But on both sides, this is, even though it's the left side of the picture radiographically, this is the right putamen, that's the left putamen. Here's a sagittal. Here you can see the different aspects where the injection is given on both sides. And it's done through this MRI compatible ventricular cannula through a syringe pump that pumps this very specific tiny amount of fluid. I tell you, the product, I was told it was a $1 million vial that I had to carry. I was designated to carry it from the experimental pharmacy to the OR, and I had to hand it directly to the neurosurgeon who put it in this device that pumped it into the patient's putamen. This is a schematic of the gene, and it's got this promoter region, and here's the actual gene product. And the geneticist can do a better job than I can of explaining the different components of this gene. 13 participants participated in the trial from 16 months of age to approximately 12 years of age. And it was pretty much split between girls and boys. And they even measured the CSF HVA level, which was low in the beginning, and it came up in time. So here's the CSF HVA, and after 8 weeks and then 48 weeks, depending on the patient, of course, they came up pretty well. This is the patient we did. Now, when she came to us, she actually lives in Thailand. Her parents said she was working on head control. That was all the head control she had at a 1.5 years of age. That was the best she could muster, okay? She's a 1.5 years of age, and that's all she could really do. Well, we did the gene therapy shortly after that point. Here she is in the operating room where her head's all marked up. Here was the MRI. Here these cannulas are basically inserted into her head. Here she's in the OR, and here's the pump that's putting the gene therapy, it's pushing it into her brain in just a few seconds of time. It's all timed meticulously. Now, we did these 18 fluorinated DOPA PET scans. So she gets, this is a baseline. March the 2nd, 2023 is 1 week before gene therapy. Her PET scans show no uptake of the fluorodopa. This is a corresponding MRI just for anatomy. It doesn't really show anything. She gets the gene therapy March the 9th, 2023. 2 months later, May 3rd, you can see uptake, that the gene therapy product injected here is taking up the radionuclide DOPA. And here's a follow-up, February 7, 2024. So now we are a year after, and it's still nice and bright. The gene therapy is working, taking up, expressing DOPA. What's remarkable, 9 months after gene therapy, she's walking. The improvement was unbelievable. It's been the most transformational experience of my career in terms of therapeutics. And Don Gilbert, my colleague at Cincinnati Children's, used that same phrase when he presented his case at a meeting we were at in Barcelona. Her family likes me to show this dress because they sent it to me at the holiday time. It was like Christmas Eve, so they make sure I show this picture. So looking at the patients that went through the trial, there's follow-up data at like 6 months and a year. And head control was achieved ultimately by 9 of the 13 kids, sitting unassisted by 4 of the kids, standing by 2, and walking by 2. I mean, it's not a cure for everybody. And it seems like the younger ones do better than the older ones. Motor milestones continued in long-term studies. Now, these come out of Taiwan, which is a different set of kids. But what they showed is that as time went on, all the way out to 72 months, 6 years, the patients are still showing improvements in head control, sitting, standing, walking, etc. And it's a one-time therapy, one and done, as opposed to enzyme replacement therapy, where you have to give it repeatedly. As far as side effects, it was really well tolerated. A lot of the patients develop dyskinesias. 3/4 have excessive movements. We take that to mean the medicine's working because now they're moving too much. And it usually occurs several weeks after the gene therapy, and it settles down several months after. So the median time to the first dyskinesia was 27 days after gene therapy, and the duration was several months, like 3.5 months or so. Of the treatment emergent adverse effects, most were mild or moderate or felt to be unrelated to the therapy. So the most common side effects were fever, dyskinesias, diarrhea, and other ones include kind of common things that happen anyway. I put together a best I could a tally of all the patients in the world treated with the intraputamental gene therapy. There's actually another approach directly into the substantia nigra, but that hasn't been submitted for FDA approval. Anyway, of this approach, 30 patients went into the Taiwan study, 13 patients went into the US study. There have been about 20 patients done now commercially because this was approved by the EMA, European Medical Association, about 2.5 years ago. So it's been available in Europe. There was a Japanese study of 8. So there have been at least 63 patients now who've received this putamental injection of gene therapy. I got the data basically from the sponsor, the PTC, which stands for something like post-translational something. That's the name of the company. The age range of the treated patients. So the study we did was 16 months to 12 years. The oldest treated patient reported was in this paper in Brain that I mentioned before from the Japanese. A 15-year-old patient who was tracheostomy and ventilator dependent. He came off the ventilator 5 months after the gene therapy. And then the oldest was a 19-year-old who was severely affected and has developed head control and partial sitting since the gene therapy. Here's my favorite picture of my little patient, partially because I myself am really a professional jazz pianist. And here she is at the piano with a little kid she goes to school with, because they go to school in their homes. And it's just incredible. I don't know if you can hear it because on Zoom, audio doesn't work a lot. But this was 1.5 years after gene therapy. She's 3 years old. And my point here is she was cognitively impaired when we met her. And now she is talking more and more and understanding more and more and showing curiosity about things like this. So her cognitive progress has been remarkable in addition to the motor progress. And those are my slides to talk about our gene therapy experience in AADC deficiency.
That was amazing. Can I ask my questions first and then folks can jump in too? In the Taiwan study where they showed the 5 years, are we to assume that there's no regression because none of those bars went down? Is that seems
No, you can't, because those bars just show where they were when they at baseline, but they may have regressed before those measurements. Those
but
are like,
it's no one lost head control, or like the first measure was 24 and it didn't.
oh, you mean during the gene therapy?
Yeah,
Yeah, I don't think there was any regression, no.
that's what those bars would suggest, right? Because the first measure, whatever it was, let's say head control, reached 24 and then stayed 24, so it didn't drop to 23. So it's not like someone
Oh,
that
yeah,
lost the ability.
no.
If they gained an ability, it doesn't seem like then they subsequently lost it.
Right. Some didn't progress.
Some
but no
didn't
one-
progress, right, sure. But if there's
Yeah.
not a progression and then regression in that specific case, I guess I'm saying hypothetically is what they're also trying to say. I don't know that. Okay, interesting.
So the gene therapy has now been FDA approved as of November. We are working on enrolling our first patient. The plan is to do in April. It takes a tremendous amount of work. I did not anticipate. The family is ready, but our hospital was going through an administrative financial process. The cost is supposedly $4 million.
Yeah.
And
Right.
that is an incredible challenge for the hospital.
And that is because of PTC, or that is because of hospital costs?
PTC.
Yeah. That's kind of what I've been hearing.
I mean, I'm not blaming PTC. I'm just telling
No,
you
of course
this.
not. Right. They have to make their
Yeah.
their money back from investing in this. Yeah. I get it.
Yeah. They must have deep pockets. They're working on a lot of conditions, actually.
Interesting. what does it take for a hospital to agree to do a therapy that's been approved? Or I guess like the folks that were done commercially, how does that work?
Well, there's a it's so expensive that there's a particularly active advocacy group, one particular mother in London who's hosted meetings on her own. In fact, that consensus meeting, she organized it. She organized a meeting in London I went to where I met Peter Clayton and Nenad Blau and all these other people, and Daryl DeVivo. I mean, it was incredible. And they worked something out in the UK that any patient will get it and it will be paid for by the National Health Service.
Wow.
Whereas
Specifically
in,
for AADC.
yeah, whereas in the other European countries, it's supposedly very challenging to get it approved.
But
Although they've done,
let's say the challenge isn't the cost from the challenge isn't the cost from the cost of the drug, but just having a hospital system do it, the administration of it.
well, the cost is mainly the drug.
Oh, okay, great. That's great.
We have a pretty robust committee that decides on genetic and cellular therapies in Colorado. I sit on that committee because we will not provide all genetic and cellular therapies because although the drugs are expensive
Mhm.
and there is some insurance reimbursement, but a lot of it ends up on our hospital, especially patients who don't have commercial insurance. And so it's a really thoughtful, long process. We actually, at the moment, would not accept a patient for this gene therapy in Colorado. We are there's a couple of gene therapies that we don't provide. But I will say that I think probably every hospital system is different. I bet what we do in Colorado is different than Boston. um, but it's something that we've constantly work on how can we provide these really rare but extremely expensive therapies to everyone who needs them.
Yeah, yeah.
Also, just want to, Dr. Pearl keeps mentioning Peter Clayton's name, who is also a hero of mine. I remember the first time he emailed me out of the blue, I actually printed the email and put it on my door. But for those of you who don't know, Dr. Clayton is also the researcher who first identified the genetic cause and the biochemical cause of PDE.
Oh, that's interesting. Did not know that. Very cool. Do you guys have any questions for Dr. Pearl that are just regular questions, like how did those families feel about having kids who underwent gene therapy? Or, like, I
Well,
don't know.
actually,
Yeah.
I could tell you a few stories that are relevant. For example, we had two families that actually came from Brazil, unrelated, they came all the way to Boston for screening. And unfortunately, the blood test showed both of these children had positive antibodies to the adeno-associated virus. And for that reason, even though they came all the way to Boston from Brazil and had the financial clearances, which included absorbing costs for incidental problems that might have developed unrelated to the trial. And we had to go through so many hoops that we had to exclude them from the trial. One of them went and found the other study group and got the AAV mediated gene therapy into the substantia nigra in the midbrain, the alternative way, and did fine. So I think these AAV mediated antibodies may be trivial, and I'm not sure what we're going to do when we find them, but it is a real consideration because it kept two families out of the trial. Another family came to us from a former fellow of mine who's in practice in North Carolina. And there's a family of two sisters. Now, they were an African American family, which is relevant in the sense that they came all the way to Boston and got screened. And one child was really severe, and I thought would be an obvious case to try the gene therapy. But when they realized this was research, for some reason they came up here not understanding that this was research and they would have to sign consent forms. They backed off and they're scared of research. It's like there's a cultural chasm there because of the old Tuskegee trials and abuses of research ethics. So that became relevant too. And then another story that I think is interesting, and this goes exactly with what Dr. Coughlin was saying, is even though we're not the only hospital that did the trial, we're the only hospital in the US, we're the only children's hospital so far that's agreed to do a post-approval patient. The other ones have
What does that
expressed
mean?
concerns, one of them totally for financial liability expressly. The other one hasn't told us, but we are the only one that's responded positively to PTC that we're going to do it because we're just going to do it.
Interesting. Who does all of the like SMA treatments? They must be hospital systems, right?
Yeah.
Yeah, not every hospital does SMA. We certainly do. I think many do.
Interesting.
Yeah, we do, but it was a big effort to get that done.
It was, we almost paused it at one point as our cost for delivering that drug impacted the care we could provide for other patients, just how we were spending. Yeah, but that is one we definitely do.
Interesting.
And then a lot of companies, like the most expensive gene therapy on the at the moment, I believe, is the one by Orange Therapeutics. What they did is they selected a few sites to be the sites that would provide post-marketing gene therapy. Um, we actually were not selected. I don't remember who was. So some companies will like work together with hospital systems and try to identify like 3 or 4 places that patients could go.
Interesting. How many total patients that are diagnosed try to receive gene therapy, do you think, Dr. Pearl, of the AADC, like, diagnosed?
I mean, since this has been approved, I've heard from several colleagues and families, probably up to, we have a list of 6. 2 of them were already patients here, and the others are new to me. And then we actually have the few that tried to go through our study that we're going to connect with. So we actually have 10 potential patients, and 3 of them have expressed a strong interest in doing it, and the others haven't really.
Do they fundraise most likely for their own cases or something like that?
I haven't gotten to the point of seeing that. I don't know. We're gonna have to see how this plays out with insurance.
Yeah, yeah. Interesting. I guess if we develop it under a different mechanism that's not a pharmaceutical company, that would reduce those costs, but it sounds like I guess maybe those are the biggest costs, and yeah, I don't know. Learning in progress. So, okay. That's really helpful. Anybody else have any other questions? Any smart questions, Curtis, that you think we should ask for recording for posterity's sake?
Well, there's this whole system of orphan drug status. I'm assuming you have that for whatever compound you're looking at. But I think that's an important
We don't.
aspect. It gives special ranking to
Right,
that
right.
drug.
We don't have a compound.
Yeah, as the group knows, I've been working with a couple of companies on a
Okay.
couple of different therapeutic approaches. And really, I'd like to believe that the reason the companies are interested is they're just altruistic and they love PDE and they want to help everyone on this call. But I think in reality,
I mean, that's one of our biggest strengths, probably, that you've somehow charmed your way in to all those.
I was actually just telling some graduate students and MD students today about how our approach to try to make the road from therapy to clinical trial as fast as possible for pharmaceutical companies so that they stay interested and their costs are low and they can make the largest profit. But really, as Dr. Pearl mentioned, the main reason we can get companies interested in our work is because of the tax benefits. Sorry, that's Christy joined my office.
Hey,
That's the end of the day. Yeah. So the current tax benefits for rare diseases, they're really important to convince pharma companies to invest in rare diseases where for like AADC deficiency, I think the incidence in the US is 1 in a million live births. Does that sound right, Dr. Pearl? I think it's 1 in
Yeah, that's
80,000 in Taiwan and about 1 in a million in the US.
yeah, that sounds right. You know, I just heard a talk today by Marshall Summer, who's just a wonderful colleague, and I'm sure you know him.
Hmm.
And he did make a point that I thought was an additional plus about rare diseases is Rare disease drugs are developed much faster and at a higher success rate than other categories like cardiac, neuro, oncology. Maybe because they're more specific, like you have an enzyme or a gene to target and you go after that, as opposed to going after Alzheimer's, epilepsy. I mean, it's just so huge. So maybe there's more, scientifically, the rationale is often tighter. But there are advantages to companies in terms of the duration required that you can get the rare disease drugs approved faster. And so if there are tax benefits and the cost is high enough that they can profit from it, I guess it works.
Yeah, that was one of the things I think they were trying to get past again, this round of funding. I managed to go to the only rare disease that day that was left, which I think was Every Life Around Us. And they were talking about passing the PPRV, right? Is that right, Curtis? Is that what it's called? Yeah.
Yeah. I will say the other thing that companies, at least the ones that talk to me about all of the rare disease work we do, is that they're really excited when there's an engaged family group.
Mhm.
And I think that you can't undersell that enough. And Dr. Pearl already emphasized like all the work that was done by that one single engaged person. But the company's always excited to hear like, hey, there's an organization, families talk to each other, they know each other. There's a place where we could talk about our clinical trial. It is a huge plus in those companies' minds. And my goal is always to add as many pluses to the columns of develop a drug for these patients.
Yeah. Yeah, but Dr. Pearl is right in the sense that when it comes down to it, right, when families have to go in and get the injection, sometimes it's a different feeling than the hope of developing the injection, right?
Well, I assume what you're looking at would be intrathecal. I'm just kind of making it maybe intravenous. I don't know.
Yeah.
But this one, intraparenchymal, deep into the brain, I think it does intimidate a lot of people.
Yeah, those pictures were
And
pretty
it's
intense.
complicated. It's high tech.
Yeah, yeah, yeah. Interesting. Well, we'll find out. Hopefully Curtis will tell us we're in March, April, in May, how we're going to try to figure out where the metabolites go. So we'll see where we need to inject it, right, Curtis? So we'll
Yeah,
talk.
we're going to continue to talk about our work. The other thing I want to highlight again is another thing that companies really care a lot about, and why we're so excited that Dr. Pearl works with us is this In Time Consortium grant all of you supported. Because what they really want to know is what are your clinical outcome measures so that we can identify whether or not our treatment works. And so I know it's not as exciting as developing a gene therapy, but I mean, it's just critical work and so important. And all of these MRI images and these other neurophysiology tests that Dr. Pearl talks about are just critical to make sure that the treatment is actually helping you and your child.
Well, I mean, if we look at the study for AADC, right, we need measures like head control or sitting, stand, things like that. So yeah, absolutely. Fingers crossed. All right. Anybody else have any questions? Otherwise, we can all go hang out for the rest of our day. I feel like Dr. Pearl, you're so intelligent. It's a little intimidating. So we apologize for no more questions, but thank you very much. We really appreciate it. This was really important for me to hear as well, and I'm sure others will listen afterwards. So thank you very much.
Thank you all. Thanks for your time and attention.
Yeah,
And
thanks.
it's good to reconnect.
Yes. Thanks.
Thank you, everyone. Thanks, Phil.
Bye-bye.
Bye.